Hello guys, I'm making this video for Lab 2, estimation of a gene and genotype frequencies within a small population. So the brief title is gene and genotypic frequency in a small population. But the purpose of this lab, first learn how to use Hardy-Weinberg principle. The second, explain the result we got from today's lab. Obviously, Hardy-Weinberg principle is the focus of today's lab. And the definition for Hardy-Weinberg principle is that without mutation, selection, gene drifting, or migration, genotypic frequency and gene frequency will remain constant from one generation to another. That's the content of Hardy-Weinberg principle. We can use this principle to calculate a pair of alleles in a gene. We can use Hardy-Weinberg principle to calculate the frequency of a pair of alleles in a gene and the corresponding genotypic frequencies either for homozygotes or heterozygous. The example we'll use is the gene in our body. If this gene is working, then we are able to taste bitterness of the chemical called phenylphylcobamide. The abbreviation is PTC. So for those people who can taste the bitterness, the genotype, which is the genetic makeup, can be written big T and big T, or big T and small t. Big T stands for dominant allele, and small t stands for recessive allele. For those who cannot taste the difference, will have genetic makeup, genotype, little t and little t. Both are homozygous. That's called homozygote recessive. The corresponding frequency we can call the small p for dominant allele. That's called dominant allele frequency, or small q for this recessive allele. It's called recessive allele frequency. Once we get those clear, we can move on. So the Hardy-Weinberg principle can be written in mathematical way. The equation first is allele frequency for dominant allele and recessive allele equals 1. If we square p plus q, the result still equals 1. We can open the bracket. Variation of that equation can be showing square p and 2pq and the square q equals 1. The reason we open it is to find the corresponding phenotypic frequency. So phenotype means the physical appearance or physical characteristic of the individual. Square p corresponds to this phenotype, big T and big T. 2pq refers to the phenotype frequency for big T and little t. Squared q corresponds to phenotypic frequency for little t and little t. If we add them all together, the result is 1. So that's the principles of Hardy-Weinberg. We'll use this principle to do today's lab. So first, we will collect the data on page 2. To collect the data on page 2, we need two types of strip. One is PTC strip, another is control strip. So it's showing here. I can get one piece of test paper and to taste it, there's nothing here. I can take another one, so there's no difference for me, indicating that I'm a non-taster with the little t genotype. Then I distribute those test papers to other students. I collect the data on table 1. Tasters are recorded using this check marker. Non-tasters also recorded. There are 2 non-tasters and 7 tasters. Those are the raw data we collected from a small class population. Next, we'll use provided data to get the genotype frequency and the allele frequency, two different populations. One is North American population, another is our class population, which is very, very small. In table 1, there are some data already provided. For example, North American population, we have phenotype frequency is 0.55, and this for taster and non-taster 0.45. So we can use Hardy-Weinberg principle to calculate the frequency for big T and the frequency for little t. I will demonstrate how to get those results using North American population. Your assignment is to calculate the frequencies in this class population. The first question is, what is the frequency of homozygote non-taster little t squared Q in class population and in North American population? So I calculate this value, North American population, then you follow my example to get the numbers for class population. So in this North American population, we already know phenotypic frequency for little t, non-taster. Know that this is squared Q equals 0.45. Therefore, Q equals square root of 0.45 equals 0.67. Use those data. You can calculate the corresponding Q value in class population. And let's move on to the next question. What is the frequency of the homozygote tasters big T squared P in class population and in North American population? North American population, Q is already known. We can use this value to calculate P based on one equation from Hardy-Weinberg principle. P equals 1 minus Q. 1 minus 0.67 equals 0.33. That's a p-value. The square p equals 0.33 squared. That's a 0.11. You can get the same value in class population. Question C is the frequency of heterozygous tasters, which is big T and little t. The frequency for this group is 2pq in class population and in North American population. 2pq equals 2 times 0.11 times 0.67 equals 0.44. So that's the way we got frequency for heterozygote taster, and the same way you can get the frequency for non-taster for heterozygote tasters in class population. The last one, when you work on get these two frequencies in two group, North American population and class population, you obviously observe the difference. The reason why allele frequency and the phenotypic frequencies in both populations are not agreeing is because the size of the population. Small size of the population usually cause large fluctuation in those two frequencies. That's all for this lab. Thank you for watching. Bye-bye.